Next up, we're going to talk about the characteristics of life. So what does an organism need in order to be considered alive? An organism needs order, response to the environment, reproduction, adaptation, growth and development, regulation, homeostasis, energy processing, and evolution. We're going to go through each one of these individually and explain a little bit more what is meant by each one of these properties or characteristics of life. So how does a cell have order? And we're calling it a cell instead of an organism because remember, sometimes organisms can just be one single cell. So in order for a cell to be considered alive, it has to have order, and we order our cell based on what's found inside of the cell. A cell has order because it has organelles. The organelles are individual functional units of the cell that are going to perform a particular function for that cell. So here you can see a list of the different organelles, and I'm going to go through each one and describe what each one of these organelles do. You should take notes on the functions of the organelles because they will appear on the test for this module. Starting with the ribosome. A ribosome is responsible for producing proteins. Later on in this section, we're going to talk a little bit about how proteins are made, and the ribosome is that organelle that is going to create proteins. You can see that we have ribosome up here, and then we have the free ribosome down here. The difference is that this ribosome up here is attached to rough endoplasmic reticulum, while this ribosome down here is a free ribosome. A free ribosome is going to produce proteins predominantly for inside of the cell. So those are going to be enzymes and such that are going to perform functions inside of the cell. While the ribosome that is attached to the rough endoplasmic reticulum is going to help produce proteins that are going to be shipped to the cell surface, so this outer surface of the cell, or shipped outside of the cell. So that kind of ties into the function of the rough endoplasmic reticulum. The rough endoplasmic reticulum is going to produce proteins that are going to be shipped out of the cell or to the cell's surface. The plasma membrane is the outer boundary of the cell. The plasma membrane is made up of a phospholipid bilayer. One of the things that I tell my biology major students is that every big word that you encounter, you try to break it down into its stems to deduce the function. Phospholipid, bilayer. So phospho is referring to the fact that it has a phosphate in it. Lipid is a fat, so it's consists of part phosphate, part fats, and it's a bilayer. Bi means two, layer, we're just talking about the layers. So it's actually two layers of fats and phosphate heads that are going to come together to create this membrane. And that'll be important later on in this lecture when we start talking about cell transport mechanisms and how things move into and out of the cell. But for now, just knowing that the plasma membrane consists of a phospholipid bilayer, and that's the boundary of the cell, that's good enough for now. Cell coat we are going to skip. That may be referring to the cell envelope, and not every organism is going to have a cell envelope or cell wall, so we're going to skip this one. And moving on to the rest of the organelles here. The nucleus. The nucleus stores DNA. That's its job, is to store and protect that DNA. The nucleolus is a dense component of the nucleus, and it's where ribosomes are made. So ribosomes are just made up of a string of RNA and some other miscellaneous items. So ribosomes are created in that nucleolus. Chromatin. Chromatin is condensed DNA. It's condensed so that it all fits inside of the nucleus. If we didn't condense it down, then it wouldn't fit. Think about taking an electric cord. Let's say we have a power cord and we were going to plug in some lights with this power cord. Now let's say we no longer needed that power cord and we wanted to put it away in a drawer, and we bundled it up into a ball and tried to shove it inside of the drawer. It may not fit that way. But if we were to take that power cord and wrap it up real tight and wind it around some type of substance to make it wrap up tight, then it would stay together and it would likely fit in that drawer a lot easier. So that's what we do with DNA inside of the nucleus. DNA isn't just loose hanging out inside the nucleus, it's all wrapped up and compacted and condensed so that it can fit and be easily accessible to the enzymes that need to access it at different times in the cell's life cycle. A nuclear pore is the hole inside of the nuclear envelope. And a nuclear envelope is the boundary of the nucleus. Just like we have a plasma membrane or a cell membrane that makes up the outside surface of the cell, we also have a nuclear envelope, and that's just the outside layer of the nucleus. Now, the purpose of those pores is to allow certain substances to pass through into the nucleus. Now, this genetic material that's inside of the nucleus is important. It's essential. We have to protect it at all costs. So the nucleus and the nuclear envelope in those pores are very particular of what is allowed to pass through into the nucleus. If the wrong substances get into there, we could have mutations, which means that the parts of DNA are mixed up so that they no longer function properly. And when we have those types of mutations, that can lead to the death of the cell. The next thing is the Golgi body, also known as the Golgi apparatus. The Golgi apparatus, or Golgi body, packages and modifies substances to be shipped out of the cell. So it packages and modifies substances to be shipped out of the cell. A little bit later, when we talk about the central dogma, I'm going to come back to this slide, and I'm going to talk a little bit about the flow of information from the nucleus to the endoplasmic reticulum to the Golgi body, and how that would produce substances to be shipped outside of the cell. The centrioles are going to be used in cell division. So later on, we're going to talk about mitosis and how cells divide or make copies of themselves, and that is dependent on those centrioles. We already talked about free ribosomes, so now we go on to smooth endoplasmic reticulum. It's kind of like rough endoplasmic reticulum, except smooth endoplasmic reticulum does not have ribosomes on the surface. And its function is to create lipids or hormones that the cell needs, and to detoxify the cell. Next item here is a lysosome. If we break apart the word, lyse means to cut up or break apart. So a lysosome is a pocket of digestive enzymes. So it's kind of like the mini stomach of the cell. So why do we need that in the cell? Well, if one of these organelles stops to function, we need to be able to get rid of it. And we can't just spit it out of the outside, because those could be broken down and used internally and repurposed inside of the cell. So a lysosome can digest those dead or damaged organelles. Also, if this cell is under attack by something that we don't want inside the cell, the lysosome has the capability of digesting it, of fusing with it and digesting it to get rid of it. If this cell wants to eat something from the external environment, we can bring it in, and the lysosome will break it down, and all of those components can be repurposed inside of the cell. So lots of different functions of the lysosome. The next one is the cytoplasm. The cytoplasm refers to the cell contents, both the liquid part and the organelle part. Cytosol, on the other hand, cytosol is referring to the liquid component inside of the cell. So cytoplasm is all of the cellular contents. Cytosol is referring to specifically the liquid component inside of the cell. Next is the mitochondrion. The mitochondrion is the energy production center of the cell. So cells can make energy without mitochondrion, but if they have the mitochondria, then they can produce a lot more energy. Now, what I just went through is the basic organelles of a typical eukaryotic cell or animal cell. Prokaryotic cells don't have all of the membrane-bound organelles. If you remember me talking about, they don't have certain organelles. So membrane-bound organelles would be the mitochondria, the rough endoplasmic reticulum, smooth endoplasmic reticulum, the Golgi body, and the lysosomes. The nucleus they don't have, so they don't have a lot of this stuff. Basically, a prokaryotic cell is just going to have some ribosomes and some genetic material, and then there's some cellular structures that help it move around and stick to surfaces and so forth. So lots of different types of proteins, and then they have just basically ribosomes and genetic material. Plant and algae cells are going to be a little bit different because they also have these storage organelles called vacuoles. And you won't need to know vacuoles for the test, but I'm just kind of pointing out some of the additional organelles that you would learn about if you're interested in plant biology. And they would have chloroplasts. And chloroplasts are the organelles that are going to go through photosynthesis. Photosynthesis is using sunlight in order to make their own food. So plant cells are a little bit different from your typical animal cell. For this class, you're only responsible for the organelles and their functions that are listed on this slide. So know the names that are listed on here, and then the functions I just went through. So make sure you take some good notes on those functions. How does a cell respond to its environment? So one of the characteristics of life is that a cell responds to an environment, or the organism responds to its environment. Well, in order to respond to the environment, you have to have receptors that can sense things in the environment. And then once you sense those things in the environment, you can adapt and change and respond to what's going on. One of the best examples that I can give is talking about hormones. So if a hormone reaches the surface of a cell, how does that hormone tell the cell to do something? Well, hormones, if you don't know what they are, they're basically chemical messengers. They're produced in one part of the body, and they have an effect on another part of the body. They usually have to travel through the bloodstream of a person in order to have that effect. Hormones are also made by plants, and some people don't realize this, but I'm sure you've done this before, where you've gotten some fruit from the store and it's not quite ripe, so you put it in a paper bag on your counter, and then a couple days later it is ripe. Well, that's because that fruit produced hormones, and the paper bag trapped those hormones inside, which allowed for more interaction of those hormones with the fruit itself and increased the aging and sweetening of that fruit. So that's an example of how plants have hormones. But with people, we have hormones that are produced in one part, and they have an effect at a part far away. And they usually, like I said, travel through the bloodstream. So how does a cell respond to it? Well, they have receptors on their surface. Those receptors can sense different changes. So in the example of hormones, here's estrogen, which is a hormone. It binds to the surface of the cell, to this estrogen receptor, and then the estrogen receptor is going to send a signal inside of the cell through a series of molecules to make a change happen inside of the cell. If you think about other ways that we can respond to its environment, if I'm cold, let's say I go outside and I'm cold, I have temperature or thermoreceptors in my skin that can sense that I'm cold. Those thermoreceptors are going to send signals through the body to the brain, and the brain is going to send signals to the effector organs, or to the rest of my body, to tell my body to do a couple things. One, it's going to try to move the blood away from the surface of the body. The blood is a big or plays a big role in regulating body temperature. So if the blood is near the surface of the body, it's coming in contact with the cold environment. We don't want that to happen. So we want the blood to stay more internal so that it can stay warmer. So blood is going to move away from the surface of the skin and go more internal in the body. Another thing that might happen is we might start to shiver. So muscle contractions generate a lot of heat due to friction. So if we're shivering, that can increase the body temperature. If we go into a hot environment, the opposite's going to happen. So we're going to respond in a different way. We are going to put the blood toward the surface of the body to try to cool it off and stretch it out as much as possible. And then we're going to put water on the surface of the body in the form of sweat. Why do we put water on the surface? Because water can change temperature faster than our skin cells can. So if water senses some wind or some air that's a little bit cooler, the water is going to cool off, and then by evaporative cooling, it's going to take the heat away from the body, leaving the cool on the body in an effort to cool us down. So how do we respond to our environment? In multiple different ways, but it's all dependent on receptors. If we can't sense it, we're not going to be able to respond to it. How does a cell adapt and maintain homeostasis? So this is kind of what I went through with the temperature example. You have a stimulus. The stimulus is sensed by a receptor. Then you have some input going into the control center. The control center is going to determine a response and provide an output and tell the effectors to produce the response. So if we go through the example I just talked about, the stimulus would be the cold body temperature. Receptors are the thermoreceptors in my skin. The thermoreceptors are going to send a signal to the control center, which in the case of my example would be the brain. The brain is going to say, hey, the body's cold, so now we have to have an appropriate response. So it's going to provide an output, so it's going to send a neuronal signal to the effectors, so skeletal muscles, and the response is going to be shivering. But how are some other ways that a cell can adapt and maintain homeostasis? So first, let's look at this homeostasis. Stasis means to stand still. Homeostasis is talking about the overall well-being of an organism, the overall functioning of the organism. We're trying to make sure that that organism has checks and balances and everything is functioning properly. Things aren't going out of control, that everything is tightly regulated and everything's functioning as it should. So to maintain homeostasis means that we have things in our environment that can change us, but we adapt and respond to those things in order to maintain an internal consistent state or a consistent internal status. So an example, let's say that it's been a really long time since we've had a cup of milk or any vitamins that contain calcium. Well, now our body becomes calcium deficient. Where do we have calcium in the body? In our bones. So the stimulus would be low calcium. There are receptors that can sense that our calcium is low, and they're going to send signals to the brain, and then the brain can send signals to the parathyroid glands, which are going to release parathyroid hormone. So the effector would be that parathyroid hormone. Parathyroid hormone is going to go to the bones and stimulate these cells called osteoclasts. And osteoclasts are going to actually digest our bone to release calcium into the bloodstream. And then that's going to increase our blood calcium levels. And calcium can go to wherever it is needed in the body. Once the calcium is released and calcium levels are restored, we shut down this system because we don't want to continue to digest our bones for no reason. So that's an example of maintaining homeostasis. There was an imbalance, our body sensed it, and we were able to correct it using an appropriate hormonal response. This happens all the time out in nature. So cells, even bacterial cells or algae or protozoa, can be sensitive to light at certain times. So if we're looking at a protozoan under a microscope and that protozoan has the ability to move, if we put it in light's path, we may see it swim away out of the light because light may be damaging to that organism. It may cause too much heat, it may cause disorder of its genetic material or disintegration of certain cell components, so you may see it swim away from the source of the light. So that all has to do with adaptation and maintaining homeostasis. That organism wants to have an internal consistency, a consistent internal environment. And in order to do that, it has to adapt and respond to its environment. How is a cell regulated? So a cell is regulated by hormones and external stimuli, and those hormones and external stimuli can tell the cell what to do. But there's also a lot of internal regulation that happens as well. There's a lot of checks and balances that take place. And one of the checks and balances would be enzymes. Enzymes are proteins, and those proteins have the ability to make a change to a substance inside of the cell without being affected by that change. So they can increase the speed of a reaction without actually being changed or affected by that reaction. And enzymes can do certain things inside of the cell and make that cell function in a particular way. One of the ways that a cell can also be regulated is by allowing certain substances into and out of the cell. So this is where we're going to take a closer look at the phospholipid bilayer, and we're going to go into the different transport mechanisms to talk about how substances get into and out of the cell. So I talked earlier about this phospholipid bilayer. Phospholipid bilayer has these little round, and they're not actually round in real life. It's just a kind of a simplified drawing of what they would look like. But this is the phosphate head. And I said it's a lipid. So these down in here are lipid tails. So phosphate head and lipid tails. Then you can see these cholesterol molecules that are embedded throughout. The cholesterol molecules are going to make this cell membrane a little bit more structural integrity. So it's going to give it some rigidity. It's going to make it tough. When we hear cholesterol, and if you think about anything you've ever heard of cholesterol, you're probably thinking cholesterol is a bad thing and we should get rid of cholesterol and eliminate it from the world in our diets and all that type of stuff. But cholesterol does serve a purpose in the cell. Not only does it help with the cell membrane structure, but it also is the basis for many different hormones and vitamins, and it's really important inside the cells. So we definitely need cholesterol. It's when we have excess cholesterol that it becomes a problem. But going back to what we're looking at here with this cell membrane, so you can see that there are some proteins studded throughout the cell membrane. Then we have this phosphate head and lipid tail kind of structure. We call it a bilayer because there's actually a top layer of phospholipids and a bottom layer of phospholipids. And then you have some protein channels that are here. So when we talk about how stuff gets into and out of the cell and how that cell might be regulated by stuff that's outside of the cell coming in or inside of the cell going out, we have to realize that this cell membrane is consistent throughout. So picture this like a wall. If you were a person and you're trying to get from this side of the wall to this side of the wall, you couldn't just walk through the wall. You'd have to find a doorway to go through. And that's where these protein channels come into play. These protein channels are like doorways. And the doorways all are specific to certain types of molecules. One molecule being glucose. So glucose is a sugar. That's a good thing. We want that to come into the cell. So we have glucose channels that will open up. They're doorways that only allow glucose to travel through. And glucose can go from one side to the other side through that doorway. There are other doorways that are regulated for certain molecules to pass through. But molecules typically can't just go from one side to the next, especially since these lipids are in here. If you've ever tried to mix oil and water, you know that that's pretty difficult to do. You can mix them together temporarily, but eventually the oil or the fat will separate from the water. So if we have water on the outside of the cell and it wants to get to the inside of the cell, it can't go through here because these lipids will prevent the water from being able to go through. So even water, to get from one side of the cell to the other, has to have a particular doorway. And it's called an aquaporin, or aqua for water, porin for a pore, and those pores allow water to travel inside and out. So we're going to talk a little bit more about how stuff, again, gets from the outside to the inside now that you have that foundational knowledge that stuff can't just readily go through. This is a wall. You can't just walk through a wall as a person. You'd have to go through a doorway. Most of our substances have to go through doorways in order to get from one side of the cell to the next. So we're going to start with diffusion. Diffusion is the transport of substances from an area of high concentration to low concentration. So the transport of substances from an area of high concentration to low concentration. Osmosis is the movement of water molecules from an area of high concentration to low concentration. So I'm going to click to the next couple slides just to give you an idea about what concentration is, so that you can understand what we're talking about when we're referring to movement of high concentration to low concentration. Another way to say this is diffusion is the movement of substances down its concentration gradient. So we're moving down our concentration gradient. So let's look at this picture here. If we look at this picture, and let's pretend that there's little holes in the wall here that's separating these two compartments, compartment 1 and 2. Now, since there's no concentration, concentration refers to the presence of molecules within a solvent. So solvent, we have water here. There's nothing inside the water. It's just water. So since there's nothing else present here, and we want to have kind of equality on both sides, water is definitely going to move from side 2 to side 1. And then we'd have equal water levels. So this is going to lose some water, this is going to gain some water, and there's going to be minor fluctuations back and forth with water until the water level is equal. But if we look at this picture, now we have some substances here. Now we have a concentration. And when we refer to concentration, we're talking about basically the space between the dots. If something is more concentrated, there's less space between the dots. If something is less concentrated, there's more spaces between the dots. So we don't care in this instance what the water level is. We care about the space between the dots. We care about concentration. So by looking at this picture, you can see that side 1 is less concentrated than side 2. Side 2 has more stuff in it. It has less space between the dots. So in order to equal the concentration, now these red things can't move. They're stuck on their sides. The only thing that can move is the water in the background. To equalize the concentration on both sides, the water from here would actually go to this side. So water would move from side 1 to side 2. Why? Well, if a little bit of this water went away, these dots would be forced to get closer together. And if water were to be added to this side, these dots would be allowed to spread apart a little bit more. So then we would equalize our concentrations on both sides. That's what cells want to be happy. Cells typically want equal concentrations on both sides of the membrane. There are some exceptions to that, and those exceptions, in order to have different concentrations of substances on each side of the membrane, usually requires energy. So, passive transport is the movement of substances down their concentration gradient, and it does not require energy. Active transport is the movement of substances against their concentration gradient, and it does require energy. We already talked about diffusion, how diffusion is the movement of substances from an area of high concentration to low concentration. And we talked about osmosis, and osmosis is specific for water. So osmosis is basically the same thing as diffusion, it's just we're talking about the movement of water molecules for osmosis. Facilitated diffusion is a little different. Facilitated diffusion is the movement of substances down their concentration gradient through a protein channel, so through a doorway. I talked about how substances can't just go through this wall. They can't just go through here. There are a few substances that can, but they're going to have to be super small, and they're going to have to be nonpolar. In other words, they're going to have to be able to interact with the fats in order to get through here. So water is a polar molecule. Small nonpolar and non-charged molecules can pass through here through diffusion. Another example of diffusion is if you were to put a tea bag in a cup of water. So as you put that tea bag in there, it's super concentrated inside the tea, but eventually the tea will diffuse out through the water. So it's moving through down its concentration gradient. Facilitated diffusion means that it's going through a particular doorway. So the example of the tea bag wouldn't work here because there's all sorts of areas where it can escape the tea bag. Facilitated diffusion would be like, let's say you're in a crowded stadium and there's one door to get out of this stadium. Inside of the stadium, there's a whole bunch of people. It's super concentrated. But as soon as you open the door, people are going to start to move down their concentration gradient. They're going to start to exit the stadium through that door. That's facilitated diffusion. So they're using a particular doorway in order to get out of the concentrated stadium or the area of higher concentration to the area of lower concentration. Now in this example, if we're talking about a cellular environment, people would only leave the stadium until there was an equal amount of people inside and out of the stadium, which doesn't really happen in real life, but if we are talking about this example in reference to a cell, we would want to make sure that there's equal amounts of people inside and outside of the stadium. So that would be facilitated diffusion. It's moving down the concentration gradient until there's equal concentrations inside the cell and outside of the cell. And facilitated means that it needs a protein channel, or in other words, a doorway in order to do that. So active transport. So for the most part, substances want to move down their concentration gradient, especially like substances. If substances are similar, they don't want to be near each other. If you have a whole bunch of super hyper positive people in a room, they may not all want to be next to each other. Or if you have a whole bunch of super negative people crowded in a room, they're not going to want to be next to each other. They want to kind of branch out and go see someone different, someone that's not like them. So when we want to do the opposite of what's natural, so what's natural would be to diffuse, to spread out, to not be so concentrated. If we want to concentrate something, a particular substance on one side of the cell or the other, that's going to require energy. And that's where active transport comes into play. Primary active transport is the use of energy to move substances against their concentration gradient. In other words, it's from an area of low concentration to high concentration. Now, why would we ever want to do this? Well, number one, what if there's a toxin outside of the cell? There's a whole bunch of toxin outside and a little bit got inside. We don't want to have equal concentrations of toxin inside and out of the cell. We want it all out of the cell. In order to get that toxin against its concentration gradient, we would have to use energy to pump it out, to make sure that it gets out. And this is exactly what bacterial cells do in some cases of antibiotic resistance. Some bacteria have developed resistance mechanisms where the bacteria can fight an antibiotic by pumping out the antibiotic out of the surface of the cell. That process, though, requires energy. So primary active transport is the movement of substances against their concentration gradient, so from an area of low concentration to high concentration. Other times that we would need to use energy, when we would want to concentrate something on one side. So when we talk about anatomy and physiology, which is a branch of biology, and we talk about nerve impulses are due to a change in electrical charge going down the neuron as the neuron is as the axon fires. In order for that electrical charge to change, we have to have a concentration of one type of ion outside of the membrane and another type of ion inside of the membrane. To maintain that balance, you need primary active transport. Other examples of why you would need energy is if you're going to move something big. So if you're going to bring something big into the cell to digest it using those lysosomes that we talked about to break it down into smaller pieces, that is called endocytosis. Endo means inside. So endocytosis is the movement of a large substance into the cell. Exocytosis, exo is talking about outside. Exocytosis is the movement of a large substance outside of the cell. So we can bring a lot of things in. We can also remove large substances from the cell. If that lysosome broke something down and it happened to be something dangerous and we don't want to reuse those nutrients, or there's toxins inside of there, then we can do exocytosis to eliminate it from the cell. So again, just kind of repeating this cell, how is a cell regulated? Typically, a cell is regulated using enzymes. Hormones and external stimuli play a role in how the cell functions, and also our transport mechanisms allow the cell to act a certain way, to have certain substances inside, other substances out, and regulate the movement of substances through this membrane. So we just covered passive transport, which does not require energy and moves down the concentration gradient, and active transport, which does require energy and moves against the concentration gradient.